Hello everyone and welcome to today's video. In today's video, we're going to be talking about blood clotting and we're going to be understanding how this process actually happens and what are the mechanisms that are implicated in this process. So first of all, hemostasis is another word for the blood clotting and or it's the process by which this happens. First of all, we have to understand that vasoconstriction is going to be taking place. Now, there are different factors that are going to be leading to that. There are many factors that are going to be causing that, including but not limited to serotonin and adrenaline, which are both vasoconstrictors. Now, the purpose for this vasoconstriction is to essentially provide time for the two remaining clotting pathways that we're going to be mentioning now. So next, we're going to be having the aggregation of platelets, which also we've got a couple of factors that are going to be implicated, including the ATP, CA2+, serotonin, thrombostatin, and adrenaline. They're all going to be working together to form a plug. This is going to be essentially reducing the number of red blood cells that we're going to be losing. And third of all, we're going to be, the process is the continuation of aggregation till coagulation and formation of fibrin. Now, we're going to be talking about that in details, and this is actually very important. So let's first start by saying that there are two major pathways that are implicated here. First of all, the extrinsic pathway, which takes about 15 seconds to finish, and the intrinsic pathway, which takes about 3 to 6 minutes. Now, also worth noting here that both of these pathways have to be complementary to each other. So essentially, the extrinsic pathway is finished and the factors are ready. It's going to be waiting for the intrinsic pathway to finish, and they're going to be working together to form what is known as the common pathway. So let's start with the extrinsic pathway first. Essentially, a perivascular tissue is going to get damaged. This is going to be releasing the thromboplastin or the tissue factor 3. Now, we have to understand here that wherever TF is written, it's tissue factor. So the thromboplastin or the tissue factor 3, it is going to be, the tissue factor 7 is also going to be interacting with the tissue factor 3 in the presence of the CA2+ in order to activate the tissue factor 10. So we're going to have TF3 and TF7 to activate TF10. This is easy. Moving on to the intrinsic pathway, we've got the Hageman factor. The Hageman factor is also known as the tissue factor 12. It's very important to remember that this is also its name. So we're going to be having the tissue factor 12, activating the tissue factor 11, activating the tissue factor 9, and then the tissue factor 9 in the presence of CA2+ and the plasma factor 3, the PF3, they're going to be activating the tissue factor 8. Now, so the tissue factor 12, 11, 9, and then CA2+ and PF3 are going to be activating TF8 in the presence of the TF9. So in the presence of TF9. Now we're going to be having a common pathway. So both of all of these factors, tissue factor 3, 8, and 7, they're going to be activating the tissue factor 10. Tissue factor 10, now this is where the common pathway starts. Essentially, this is taking 15 seconds. This is taking 3 to 6 minutes. And now we're going to be having the tissue factor 10. This tissue factor 10 in the presence of the tissue factor 3 that we mentioned earlier, the thromboplastin, the plasma factor 3 that we also mentioned earlier, and also CA2+, present also here, they're going to be activating the prothrombin activator, also known as the prothrombinase. Now, this is going to be a cascade of events. This is going to be activating the prothrombin, which is also known as the tissue factor 2, and this is going to be converting the prothrombin into thrombin. And here there is a negative loop, negative feedback system in order to just, in case the levels of the thrombin were very high and this was not required anymore by the cell, it will simply stop. So basically, the thrombin is going to be facilitating the conversion between fibrinogen into fibrin, and then fibrin into fibrin polymers in the presence of factor or the tissue factor 8 and CA2+. So essentially, this concludes this episode. And if you have any questions, feel free to drop them down in the comment section below. Thank you for watching.